Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. In podcast number 12, I talked to Abrama Blackwell about modeling of signal pathways inside neurons. For example, modeling of the intercellular cascade of molecular reactions behind strengthening of synapses. This kind of modeling is difficult since so many different molecule types are involved and lots of more or less uncertain model parameters must be specified to run the models. Imagine if you're even more ambitious and want to simultaneously model all processes in a cell, not only signaling pathways, but also the consumption of energy, production of RNA and proteins, and everything else that is needed for the cell to be alive. This is exactly what today's guest, Markus Covert, has done. First, in 2012, he and his group at Stanford University published such a whole cell mathematical model of the bacterium Mycoplasma genitalia that went through the life cycle of real bacteria and, like the real thing, split into two cells after about 10 hours. Then, in 2020, he and his group published a similar type model of the much larger and complex bacterium E. coli, the favorite bacterium to study for microbiologists. This E. coli model incorporated 1,214 protein-producing genes, about half of the total number of genes in the real bacterium. The model included more than 10,000 mathematical equations, with about 19,000 parameters estimated from reading thousands of experimental papers. This was truly a Herculean task. These whole cell bacteria models have done much more than just reproducing experimental data. They have also come up with new predictions that has been confirmed in later experiments. I learned about this work in 2019 when Markus and I attended the same meeting at the Allen Institute in Seattle. I was very impressed not only by the scientific results, but also about the guts that he displayed when deciding to pursue this very ambitious whole cell modeling project as a young professor at Stanford. I don't think there was a lack of cautionary warnings from biologists against pursuing this project at the time, with arguments such as cells are too complicated to be modeled in this mechanistic, physics-like way, there is too little experimental data available to develop and test the models, there are too many unknown parameters, and so on. Nevertheless, Markus was not dissuaded, and the project paid off in the end. In the podcast, we talk about why first the Mycoplasma bacteria and later E. coli were used as test organisms, the different cellular functions that are needed for a cell to live and procreate, the key strategy of organizing the whole cell model as a collection of somewhat independent modules, how the dynamics of the different modules was modeled using different mathematical formalisms, how model parameters were extracted from comprehensive readings of literature, what kind of experimental data was used, how the model and modeler parameters were tuned to make the model predictions agree with experimental data, and finally how new predictions from the resulting whole cell model was tested against new experiments. We also talk about how this whole cell modeling work has been received in the largely experimental microbiological research community. As we know from neuroscience, not all experimental neuroscientists are convinced about the usefulness of computational neuroscience. Finally, we discussed the future prospects of whole cell modeling, maybe even with similar modeling of multicellular organisms, and also the possible use of AI to aid the effort. While the topic of this podcast is somewhat outside the core of computational neuroscience, I would think it nevertheless should be of interest to many of the listeners. I attended a talk by John Hopfield in Sweden many years ago, and I remember he started the talk with saying something like, "In computational biology, we have to learn to compute without knowing all the numbers, and that is very difficult." This difficulty is shared between whole cell modelers like Markus and computational neuroscientists modeling signaling in neurons and neural networks. So I think our communities can learn from each other. Also, I think the boldness of Markus to pursue the first whole cell modeling of a living cell should serve as an inspiration for us computational neuroscientists to also be bold and dare to go beyond our comfort zones and pursue For example, modeling of information-rich, complicated neural systems, where both the model and the data it tries to explain are unwieldy. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking. For example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I now also have established a YouTube channel called Theoretical Neuroscience Podcast, where you also can watch the videos. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community on social media or otherwise. Okay, here is Markus. So, welcome to the Theoretical Neuroscience Podcast, Markus Covert.
Hi.
Hi. That's a good start. Okay. Anyway, this podcast is about mechanistic modeling of cells, and I remember, I think we met like in 2015 or 18 or sometime. A few years ago, where you gave a talk at Allen Institute about this first whole cell model of a bacteria. I guess it was this really seminal paper in 2012, where and then you made you and your group at Stanford made this like first whole cell model of this mycoplasma genitalium. So that's so for those of us who are not so familiar with all these small bacteria, what is this bacteria, and why did you start with that?
Yeah, great question. And it's really great to be here. Thanks for inviting me. Yeah, the mycoplasma genitalium is it's just the simplest culturable bacterium. That was the idea. It had the fewest number of genes, and kind of like by analogy.
Well, the culture will mean that you can have it in the lab quite easily.
Yeah, you can grow it in a lab. It's very fragile. Like at the time, I had some students who were worried about working with it, and I said, well, yeah, it's an STD, but literally, you would have to pour it down your pants and sit in a hot tub for this thing to take any kind of, this is not something you need to worry about catching in the lab. It has a very strictly defined media, and if it doesn't get that media, it pretty much just dies, right? So it's very fragile, which is one reason why we moved to E. coli as being a much bigger challenge. But I always thought about our work as kind of following what for me was the greatest source of inspiration during grad school, which was the genome revolution, right? So my vision was simplest mycoplasma. It was in the first 2 sequence. Then to E. coli, which is 10x in the genes, but like probably 50 to 100x in the molecules, much bigger. And then move on to, let's say, a yeast or to a mammalian cell. Of course, as I don't know if we ever talked about this, but I'm now convinced that we could probably skip yeast and go to a mammalian cell. And if I were to choose a cell, it would probably be a beta adrenergic neuron. So heavily influenced by Paul Allen. So we can add that to the discussion later. But E. coli has been a gold mine, and I feel like we're in this transformative place with E. coli. But mycoplasma was a lot of fun.
Yeah, because that was sort of like it was almost like the hydrogen atom of systems biology, right? It had, for one thing, it has about 500 genes. So which genes?
525.
Okay, which encodes proteins. Okay, and so, but that's not the whole genome. So what does the rest of the genome do?
There's very little other material, actually. Mycoplasma is so evolved down. In fact, one of the really interesting things, it's come up with these ways to be parsimonious that are fascinating. So for almost every organism on the earth that we know, they have an NDK gene. It's like a kinase that basically transfers. Think about it as a currency converter, right?
Mhm.
Dollars to euros, or in this case, GTP to ATP, that kind of stuff. It's essential for almost everybody, but not for the simplest organism on the planet. And the reason is that basically to remove that essential gene, it evolved a very kind of promiscuous glycolysis that can use any of the currencies, and as a result,
Glycolysis is a way to eat sugar or whatever, or
to go from glucose to metabolize glucose all the way down to pyruvate.
So that's part of the metabolism.
Yeah,
But
and
this is
typically
a neuroscience community, so we are not
right.
we are not so
So
skilled in these basic biological things.
yeah, usually we think about it as generating ATP, right? The energy required for everything else to happen. But in mycoplasma, in this very simple cell, it can generate ATP, GTP. So it's not only like an energy generator, but it's also kind of a currency converter in the same step. And so that enables, mycoplasma presumably to go from 526 genes to 525. So you can just imagine how much it has reduced this genome.
Because I think, this was sequenced, as I said, you sort of mapped out the gene structure by, this J. Craig Venter, right, and his team in 1995. And then he also used this to make even simpler artificial life, didn't he?
So in fact, that was how I got into that exact work.
Okay.
Identifying the minimal gene component for a cell. That came out in Science probably in 97 or 98. 98, it must be 98. And that was the second genome. And so they did the world's simplest theoretical study that ever made it into Science.
Okay.
They took the list of genes that was in Haemophilus influenza. They took the list of genes that was in...
What is that? That's another virus? Bacteria?
No, that's a bacterium. It was the first sequence.
Okay.
And the second one was this mycoplasma. And then they took the intersection of the gene lists and they said, aha, the minimal set of genes to obtain life. And that made it to Science.
Well, I think it's a wonderful idea.
It is. Unfortunately, it nowadays we would know, thanks to systems biology, systems approaches, that of course there are going to be all these nonlinear interactions between those genes. And so
but it worked.
That's why so then they started making, they developed all this incredible technology, like brilliant Danny Gibson, John Glass, all those guys, Clyde Hutchison. Just an incredible team. Developed all these methodologies for synthesizing, chromosome scale amounts of DNA, which was incredible, and then transplanting it into a cell. And so what that work led to was the ability not necessarily to make something dramatically smaller than mycoplasma genitalium, because but they were able to get mycoplasma, I believe it's mycoplasma pneumonia, if I remember right. And they've reduced it down to a synthetic strain that's, I don't remember the current gene count, but it's probably roughly in the ballpark still of the mycoplasma genitalium, but it grows much faster.
It's like 400 something, or I don't know.
Maybe, what I had in my head was 470.
Yeah, that's I don't know. So that sounds
it took them a long time, and I think exactly because they started, wondering if they should just do an end run and actually just try that from scratch. But of course, you could have a situation where, let's say you have 10 genes, right? And I delete each one singly, and I find out that there are 2 genes where if I delete them singly, the bug can still live. So I delete them both, but then the bug dies because they are both encoding the same function or a redundant function.
Yeah, exactly.
So it's very hard to know a priori because of all those nonlinearities. But they've been amazing. Like, what they've done has been amazing over the years, and they continue to make progress. And they've identified a number of truly essential genes that nobody has any idea what they do. So that's pretty incredible.
So now we're going to talk about, of course, your whole cell model, which came like whatever, 15 years later or something. But first, we need a little sort of crash course for the neuroscientist about like the basic function of the bacteria, because of course that's eventually all these functions that you have to put into your model. So I guess the first thing is metabolism. What's that about? Why does bacteria have to do metabolism?
Yeah, sure. So let me add one great little segue, which is that when they published that paper, I think it was there was an article in the New York Times, and Clyde Hutchison actually said, I was a brand new grad student. So I was reading about the genome revolution and I was like, this is it. This is what I want to do with my life, right? Like but then at the very end of the paper, there was this quote that said, the ultimate test of being able to understand a cell more than being able to build one would be to build a computer model of a cell, because that implies a different level of understanding of how it works. And literally, I would tell myself this quote every day for like 20 years, right? Like I was obsessed with it. And so you're exactly right. Then what we had to do is start thinking about how to do it, right? How do you actually go about and do something that, people were calling like the grand challenge of the 21st century. So I at the time wasn't sure we were going to do it, but I was working in Bernard Paulson's lab in San Diego. And at that time, he was the world leader in modeling metabolism. And as you say, what is metabolism? Metabolism is basically the system of biochemical reactions, right? Catalyzed by enzymes, usually, that basically take you from the nutrients that you eat to all of the energy and other needs that you have to be able to grow or sustain your life, whether you're a human or a bacterium. Metabolism is a necessary requirement for life, right? And it's about as fundamental as it gets, because it's kind of the engine generating everything that you need to survive and live in a healthy way.
Sort of like and as a physicist, I would say, I mean, it's like the thing that sort of makes you extract free energy, sort of from the nutrient that comes in, right? And then excreting waste. And then, of course, you said that the enzymes are important, and enzymes are like a type of protein, right? And then another thing you need is sort of like the genes. I mean, right? So what is the gene system in the bacteria?
Right. Well, this gets into all the nitty-gritty details of modeling already, because enzymes are usually a protein, right? But there are other things that can catalyze reactions like RNA, et cetera. And some reactions happen spontaneously. And what whole cell modeling is, I can give you the definition right now. It's just to try to come up with a mathematical representation. It's the process of trying to come up with a mathematical representation for every single thing that happens in a cell. And so that means, there are all these obscure details about how cell works that most people never think about, even people who are cell biologists, right? Because they're just obscure, they're rare, but for us, they're really important because we're trying to take everything into account. And maybe in a physics term, we're not just interested in, it's like the van der Waals attraction. Like a lot of small attractions make a big difference. And so a lot of small behaviors also make a big difference. So you've got the proteins, you've got the RNAs, which are useful for all kinds of things. And it feels like we're constantly finding new uses for RNAs that are being discovered.
But the standard, I mean, the most sort of what is called the standard story, no, what's called the hypothesis, whatever. Also called this thing
the central dogma.
Central dogma, exactly. That is sort of going from DNA.
Right. Since we have a new pope, it's time for us to talk about central dogma. Yeah. But maybe it's new dogma we need to talk about, because the central dogma, as coined by Francis Crick, is that information flows from the DNA to the RNA, right, to the proteins, right? And so the DNA is transcribed to make RNA. The RNA is translated to make proteins. Nowadays, we know there's all kinds of other stuff going on. Epigenetic regulation at the DNA level, right? In the immune system, some immune cells will even lyse and use their DNA as like or even not lice, and use their DNA as a net to trap stuff, right? So that everything has
really?
more uses than you would think, right?
Okay.
And so the dogma is helpful, but it's turned out to be dogmatic, right? So it's not totally useful. But in our case, the fundamental part is we're trying to then map, you're exactly right, all the genes to whatever they encode, be it RNAs, and then we're looking at the non-genetic material as well to see whatever it encodes and what we need to do with it. And then we try to represent that mathematically in the most appropriate way possible. And then it's a matter, and that's usually different for different functions. Like the way that we represent the transcription into RNA is very different from how we represent metabolism.
I mean, we're coming to that. I just want to get the basic biological process. No problem.
Yeah.
So they have metabolism, and they have like the genetic thing, DNA to RNA to proteins, and then you also have this sort of decay and recycling, right, of RNA and protein. And you don't talk so much about carbohydrates, but that's maybe there's not so much sugar or things.
It's covered in metabolism. Or do you mean on the outer membrane? It depends on what you mean.
I don't really mean anything. I just sort of from a biological sort of like player, but like actor parts. I learned about like protein and DNA, RNA, and carbohydrates.
So lipids, of course, are a huge deal because they are protecting the entire cell and kind of regulating what gets in and out. And they also play a big role in energy generation by letting certain stuff in and pumping certain stuff out. And then, and so that's lipids. And then you have things on the outer membrane that are also interesting for recognition or interaction with the outside world, whether it's proteins that have made a channel to let stuff in, or force stuff out, or you can have, things on the outside, often sugars, that are just recognized by other stuff or stick to stuff, so there's all kinds of stuff going on. Typically when we talk about the small molecules and how carbohydrates are used, that's all going through the metabolic network, right? So it's being processed and turned into the other stuff. What else is in there? I mean, one thing that, go ahead.
No, I mean, you also, because I've been reading your paper. So you also, this thing of there's damage. I mean, you have to repair the DNA when it gets damaged, right? Some kind of, first aid.
What's funny about that for the simple model that we made in 2012 is that to make a model of DNA repair, we thought, it looked like it wouldn't be too hard. But then once we made the model of repair, we thought, we forgot we have to make a model of damage. And that's harder because it goes into physics and how different wavelengths of light are damaging DNA in different ways. And so, sometimes you have to do exactly that. Like it's not genetic at all, but you have to incorporate the physics of the real world that it lives in.
And of course, one target was sort of to go through a whole life cycle of this bacteria and actually make it divide into two, right? And then you also need to include that process, something called a formation of a dividing ring, some kind of thing that cuts off cuts the cell in half.
So the cell kind of grows out as like a cylinder that just gets longer and longer, and once it hits a certain length, it a bunch of little filaments, little kind of rods, little chains of protein join together, and they form this ring that kind of contracts and cinches off until they pinch all the way, and then these two cells are divided, and each of them can start their own life cycle again. But yes, that's this gets into the modeling a little, but the most interesting insight that I had that really enabled whole cell modeling was to realize that it might be better to model a single cell instead of a population of cells, which I think really runs against what prevailing wisdom would have been, but turned out to be correct for a number of reasons, which we can talk about later.
Okay. Why is that? I mean, it's just like it's certainly a smaller system with one rather than many. You would think, like one hydrogen atom is simpler than two hydrogen atoms.
Yeah. Well, I think you
Well,
get a few things. You get, we used to say what you get is molecular closure and temporal closure. So, molecular closure means, I think if I were to write a system of ordinary differential equations, it would say something like, oh, transcription factor X binds to every gene with a certain, if you have, it will go out like 0.015 of it will go here, it will go here, it will go here. And so parameterizing that or figuring that out is very difficult, but we benefit from the fact that if you have in a single cell, you have some transcription factor, let's say, or have something that was going to bind to the DNA, and it has 3 places it can bind, it will only bind to one of those 3. So there are 3 options. It can, well, 4. It could bind to one of those 3 or bind to nothing. And so, and it will do one of those things. And so you don't have to consider every single crazy thing that could possibly happen. You can just say, okay, it is going to bind to one of these things. It has a specific place and it will go to that specific place. And then you know where it is. And once it's on the DNA, you know how fast it goes, right? So you can use a lot of the single molecule biophysical measurements that have been generated over time. So the thing about molecular closure is, just think about it this way. There's a finite number of molecules you have to deal with, and every one of them has a finite number of things that they can do. And you can basically make a simulation that will run through those steps. Temporal closure goes back to what you were saying before. The other thing that is really powerful with a single cell, that it has to double everything so that it can, it's not strictly true, we now know, but at the time we were thinking it has to basically double everything so that it can divide in half and have 2 halves and start again. So that was an incredibly powerful, when you think about constraining the parameters that are necessarily involved in a simulation like this, having that requirement just collapses the parameter space.
now to sort of making the model, I think there's, you mentioned in your paper this Harold Morowitz,
Yeah.
was, I mean, almost 20, well, in 84, already had a plan for this whole project.
Yeah. The 3 most interesting, 4, there were 4 people that I followed a lot. And of course, I was in the Bernard's lab, and that was amazing.
Yeah.
So that's like the first person.
Yeah, actually, I read a bit in his book. It's quite excellent, right? I think, yeah.
Yeah, he has a number of books. They're all great.
Okay.
And then Brenner and Crick. So Francis Crick and Sidney Brenner in 73, they called for a complete solution of E. coli. It was not quite with the same, it's fascinating. And if you look at the Cold Spring Harbor archive, you can actually find a piece of paper where Sidney Brenner, they called it Project K because E. coli, the strain is called K12. But then he had a Project K, L, M, N. So K was E. coli, L was bacteriophage lambda, M, I think, was the mouse, N, Sidney Brenner, so it's the nematode, right? C. elegans. So anyway, he had this whole alphabet of models that he wanted to solve.
Okay.
And then but there weren't a lot of details there. And then you had Morowitz with this incredible statement that, like, anything that you can run in a simulation, you could also, any experiment that you could possibly do, you could also run in the computer. And he had this thing that, again, another quote that I basically memorized, which is that, finding the difference or the discrepancies between those 2 is how we should be learning new biology. And that is exactly how we do it.
Of course, this thing of sort of, if you understand it, the only way to test whether you understand it is to build it, meaning in a model.
Right.
That's like a thing, right? So this idea is sort of quite well established in physics, where of course the complexity is much less.
Yeah.
So it's an easy program to pursue. But at least this idea of making mathematical models, sort of that is sort of
Yeah.
prove that you
And
understand.
I look to those things all the time, right? I look a lot to weather forecasting.
Yeah.
In 1900, in the early 1900s, the first volume of Science, a meteorologist called for the consecration of the physicist and the mathematician to this work, right? So that real progress could be made, and of course they did that over 100 years, and I would say, and then the other is astronomy, right? Like somebody looked in the telescope and was checking all those orbits, and they realized there must be another planet, and they kind of picked, okay, look now, right? Like right now here, and they found Neptune, right? So it's kind of an amazing. So that's definitely the model that I consider when I'm thinking about this stuff. So yeah, then Morowitz said that, and then there was this guy at Cornell, Mike Schuler, who I would say is kind of the grandfather of the field, where he was really trying to do some very amazing modeling without the genome. So it's kind of more like a cell physiology, but with increasing detail. I mean, it's beautiful work.
Yeah.
And then right, and then eventually, then you had Paulson, and eventually we showed up, and fun.
Because
Fun
now,
field.
okay, so back to the model, because now I read that, I mean, there are 500 and whatever 25 genes, and I think it says somewhere that it's only 284 genes are essential. And 117 is non-essential.
I don't
think maybe,
remember the exact count, but it's
but it's
actually mostly essential, and a small number. I don't remember if it's 100, but it could be like around 100 that are
non-essential. Yeah, I think I
non-essential.
just took the number from it was like a 20s, this Dream 8 paper challenge.
I
I
see.
don't know, but
Yeah.
anyway. But so in your model, then you essentially you took into account essentially more or less all the genes.
All the ones. So I would say it was a complete functional representation. So not all the genes are known. Even for the simplest bacteria, we don't know. Even for E. coli, we don't know what over 1,000 of the genes do.
Oh, okay. So, and of course, then if you don't know their function, it's difficult to include in the model. But
yes, but we do include them. So importantly, if they are made, they exist, right? They are taking up space in there. So if we deleted them, they still would have some function, right? Probably or not function, but they would have a very small effect, because their more energy would be given to the other protein expression, right? But we include every function that we feel like is well enough characterized that we can include.
And then you start. I mean, you assume that the membrane is there, right?
Yeah. We include the membrane. Super
Well, I mean, it's
important.
not something you don't model the formation of the membrane. And
Yeah.
then
So here's a distinction, right? For some people, when you hear whole cell simulation, and I would say this belongs to the field, right? People, we started using this term. Other great groups are also using this term to mean slightly different things. And I think there's this idea of an all atom, like every single thing included, We don't do that for a variety of reasons, but the biggest one is that I want these to be really functional, right? I want to be predicting the outcome of real experiments. Our lab is fully functioning wet lab. We do our own experiments. And I want to find, my biggest goal, right, is really to find Neptune, right? I want to find biology that people have never seen before. And so that kind of thing would be hard to do in an all atom representation, which tends to be much shorter, much more limited in volume, and often, surprisingly, not that functional.
Yeah, no, absolutely. And I think I was thinking more that actually the membrane is something that you assume the membrane, you assume like the bag of the cell and a certain start volume, right? And then you put in these different, I guess, different modules. That was also like a key idea,
Yeah.
right? That you said you were inspired from a course you took in chemical plant modeling, right? For you.
Yeah, that's right. Wow. Yeah, it's amazing that you knew that.
Wow.
So yeah, I was a chemical engineer as an
Okay.
undergrad. I really did not enjoy that major, mostly because we were so focused on petroleum and distillation columns and stuff like that. But our capstone project that we had to do was fascinating to me. So I worked in molecular dynamics with a brilliant professor, Richard Rowley. And then for my capstone project, I had to design a chemical production plant. And so we had to design all these towers, and each one of them worked independently, but then they came together and were integrated. And that's essentially how the whole cell model works. I realize now, I didn't even quite realize that for years after we had done it, but then someone reminded me of my traumatic past in chemie.
Yeah. So then you essentially had to decide on these different modules. As I said, it's 28 distinct modules. And what are these modules? They have like different functions, right?
That's right. They're different functions. So like one of them is metabolism. One of them is transcription. One is protein decay, right? So we all these kind of broad classes, and we fight a lot about that. That's where modeling becomes more of an art than a science, because you have to sit around and for each one you have to think, okay, well, how should we group this up? Is this big enough or too big? And then we have to think, okay, what is the representation we are going to use for that module? And that's very subjective, because usually you're looking at data, because in some cases you have single cell data with a lot of time resolution. It's amazing. In fact, we've generated a lot of what I've done on the other side of the lab with mammalian cells is to try to generate ways to measure things that are more single cell and more dynamic. But in other cases, you might just see, you might be reading a paper and you might see an X pointing to a Y in one of the figures, and there's a question mark there, right? That's what you've got. That's all you know. And so then you have to be much more qualitative, right? Doug Laufenberger has had really interesting things to say about just how depending on the level of quantification you have in your measurements, you have to change your level of abstraction in the modeling method. So this is one thing that really helped us. I think where we were able to go a little further than others was, I think most people at that time were thinking of using one method to cover everything, whereas we were able to see, if we just use the most appropriate method for each of these things, and then come up with, instead of new methods to represent the whole thing, we come up with methods to integrate. So most of our work was an integration of different methods. And so you can think about our model as like a patchwork quilt, right? As just a way to bring a bunch of stuff together. And that turned out to be the secret sauce of how it works.
Because then you could also take advantage of models for modules that had been used before, maybe, or formalisms. So you didn't have to reinvent all the wheels.
Right. We didn't. In fact, that's exactly right. And we prefer in the E. coli model for right now, for example, which I hope we'll get to talk about, because that's the real, that's the amazing. But that thing has probably between 19 and 20,000 parameters that we took from the literature. And of those, in our science paper, probably 50 are fit, right? The rest are just used as is. And if we find models that exist, we really try to use them. I want the model to be a community achievement, the way that weather prediction is a community achievement.
Because one thing, because when I sort of think about modeling things inside cells, I think about sort of like some of these what we call mass action equations, and like sets of ordinary differential equations, right? But it's not. That's not the only thing you used, right?
That's right. So sometimes we use stochastic simulation. Sometimes we use probabilistic models.
Stochastic, that could mean like an ordinary differential equation with a stochastic source term or something, right?
Yeah, but what I'm talking about is more like a Gillespie simulation, where again, it's leveraging the discrete nature of the molecules involved, right?
Exactly.
So it's not adding a noise term to the ODE. But we do that too, I'm pretty sure. So we do probabilistic, hidden Markov models. Like there's a whole bunch. There's Boolean models. There's a whole bunch of stuff that goes in the sandbox. They all have to play nice together.
But then, of course, we also had to decide on. I think you mentioned how to. I mean, it's the typical variable concentrations of the molecular components. Is that sort of like the?
That's a very common variable. We often, depending on what we're using, like ODEs versus SIMS, right? We might go with counts. We might go with concentration.
Okay.
For other things, it might just be location on the genome or on the chromosome. Okay. For other things, it might be compartment that it's located in, like a membrane or between the two membranes, or So there are lots of different variables that come into play. Honestly, it's a pretty fascinating thing.
I think I really like this pragmatic approach. Just doesn't get stopped by some kind of philosophical issue. You just sort of hammer along.
Yeah. Do you know the movie? Maybe not everyone here will know, because this is like very U.S. almost propaganda movie, right? Apollo 13. Have you heard of that movie?
No. I think most that's certainly popular in Europe also.
Yeah. So I would say, I always tell my team, like every engineer, it's a requirement that their favorite movie is Apollo 13. Okay. And their favorite part of the movie is required to be, there's a part in Apollo 13 where they basically have to rescue this spaceship, and a true story. And the way they have to rescue it, there's this part of the spaceship that's broken that's going to enable the team to have enough oxygen. And so back down on Earth, they bring this pile of junk, which is a bunch of stuff that's in the rocket that they could potentially use. And they tell them, you have to use this somehow to fix the ship. And I tell them, the reason why, this is where engineers shine. Because if you were to ask a pure mathematician or a theoretical physicist how to fix it, they would say, it's impossible. It's impossible to do it because there's no solution, right? And there isn't any solution. But this is where an engineer, the mindset is helpful, because they can say, yes, there is no solution possible, and yet we have to do this. And so we're going to do it, right? And so that's literally what I told my team at the time. I said, everyone says this is impossible. I'm not asking you to find the perfect solution for some impossible thing. I'm simply asking us to come up with the best solution, right or wrong. And of course, it will get better over time. So that's like I would call that the pragmatic engineer's approach.
So the perfect should not be the enemy of the good. Right?
Yeah, exactly right. Yeah. That's it in a nutshell.
So you do also use like statistical models as part of this?
Yeah, you mean like statistical mechanics?
Well, I'm sort of thinking of sort of, if you look at sort of the Hodgkin and Huxley model, of course, celebrated for the single neuron dynamics, the input, the ion channels are still modeled sort of statistically, just sort of like with fitting formulas, essentially. So you have these models which are not mechanistic, but then are used as to model input.
I see. Yeah, so sometimes if there's just a relationship that's known, we would totally use it. Yeah. We're not finicky. We're not picky eaters. we'll eat we'll eat lutefisk if that's all we get is experimental lutefisk. We'll eat it.
I used to be a rock climber, and then there was this saying that it's too cold for ethics. A little bit related.
Yeah, no, I love it.
Yeah, so I like that approach. So anyway, but then you have these modules, and they are modeled sort of like different kinds of techniques and different kinds of mathematical whatever equations, and then you need to come, because I guess the key thing here was that you separated time scales and said that the modules sort of have like a fast dynamics inside, but then they communicate on a much slower dynamics. Maybe like, I think you said the time resolution of 1 second.
That's
I guess that was an important thing, both conceptually and practically.
Yeah. It essentially enables us to transform the problem into a numerical integrator problem, which has been well solved by many people. It's just a way of representing dynamics of a system in time. So yes, and the key, this is actually one of the questions that I used to dread when I would originally give this talk, which you'll see why in a second. So the idea is that of course, everything is happening all at once in the cell. But what if you could say for our modules that they're essentially independent for 1 second? Like they interact with each other on a time scale of 1 second. That it enables you to run it essentially as 28 models in parallel, or in series or in parallel, but always coming back together at a second. And that is a really, really critical assumption, because otherwise we couldn't have 28 modules, right? They would all have to be running with each other at every single moment. It turns out not to be a big deal as far as, taking the assumption does not break the system, and it helps. It is essential to having the system work. And I would say the reason why it's an embarrassing question, people would always ask me, what's the programming language you use? And then they would also always ask me, how did you choose the 1 second time step? And the answer is, back when I was starting to do this, everyone just thought I was crazy, right? No one wanted to take it on. So I was doing it by myself. And so I was just mocking stuff up in MATLAB. So I used MATLAB for this. And then, I was a busy young professor, so I didn't have any time. So I would have all these lists of parameters, and they were all per second, and I didn't want to multiply by anything, so I would just take them as 1 second. So it's like the worst reason to choose the separation scale.
That's
but
true.
Now we've tested it, and it really is between 1 and 2 seconds is the right place to put it. So that's the good news.
I guess also, if you think of I don't know if I should make a cell and with different modules and functions, that you need some kind of separation of time scale. If not, things just get completely bungled up.
Yeah,
so that's
totally.
So then you have these so you have these modules, and each of them must then have some kind of representations of the things to communicate to the other modules, right?
Yeah, they all have like a very thoughtfully designed input and output. And for the most part, there are exceptions to the rule of independence. The big ones are ATP, which is used by everything. And so we kind of have to do a pre-allocation and a post-allocate just to smooth that part over, because ATP really does tie everything together on short time scales. So there's this chemical connection that is a violation of the assumption. And then the second thing that's a violation of the assumption is that there's a lot of this. I knew about the ATP, but I was surprised about the physical interaction, which happens on the genome, so on the chromosome. So, the entire chromosome space gets visited very rapidly. You could think of the DNA as just a molecular ecosystem where all these things are running around. You see it in the paper. We estimate tens of thousands of interactions that lead to some protein being forced off. And that's astonishing, really. Like that's its own physical problem that's really fascinating. But that interaction also we had to kind of think about and account for as a potential violation of the independence of the module.
So then, so you had all this state. I guess it's sort of like this state variables that were these cell variables that was sort of like metabolites and concentration of this and that and RNA, and that was sent between this module. And then, I guess, did you assume that it was like well mixed? You didn't take into account diffusion explicitly, did you? Or
yeah, so we have compartments, but those compartments are for the most part well mixed, again with the exception of the DNA chromosome, right, where things really are. And so the DNA chromosome and every individual mRNA, because ribosomes also bind to the specific places.
I see. So they are they have some spatial position.
But it's more like a 1D spatial positioning, because it's along the strand. if that makes sense.
Yeah. There's no osmotic flow of water and stuff that you didn't.
No, we don't do a lot of the detailed membrane stuff, and even in E. coli, that's something that's going to be big and interesting. It's also related to the charge balance, which is why we've been reluctant to take it on. That's a very intense. Talking now to a neuroscientist, right? That's the charge. The charge is a big deal, and it's so much more dramatic than you'd think. Because like DNA is highly charged. Most of the proteins have something going on, right? There's it's a really complicated system, and one that we could definitely use help on. But it's in our sights.
I actually did my master thesis work on the distribution of these counter ions, positively charged counter ions.
Oh, really?
Yeah, that was back in the. Well, this was my first paper in Journal of Chemical Physics back in the.
I'd be more than happy to offer you a postdoc.
That sounds very tempting, actually. So anyway, so then you end up with this. Of course, now it's bigger with E. coli, but with this mycoplasma model, then how many equations did you end up with, or ballpark?
Of what? Sorry, say again.
How many equations, if that's sense? I mean, you had like a few hundred equations, or
what do you mean by equations?
Yeah, like mathematical equations.
I mean, I see. Yeah. Sorry, equations. My bad. Yeah, sorry about that.
No, it's probably the Norwegian accent.
I love it. So equations is hard to say because, but I would say, you could estimate it. You could estimate it probably at something like, over 1,000. Maybe 1,000 to 2,000.
And then you end up with, I think, about 1,700 model parameters that you have to deal with.
Right. And so almost none fit. That's the other crazy thing about it, because you have to, saying that you don't fit them, I think shocks a lot of people, because it is extraordinarily difficult to fit even small systems of parameters. And so for that reason, we often try to represent, like Bernard does, right? I would say what Bernard does in some ways is representing systems of ODEs as optimization problems. And so we've had to be more sophisticated than the original metabolic models.
But, so what do you mean by it's an optimization problem?
So you could say, for example, let's say that you want to use all the parameters in a set of ODE, ordinary differential equations, right? You could either write the set and solve the set explicitly, or you could write that as an optimization problem where you say, I want to minimize the change in here's my set of equations, and here are the parameters that I want to use, and I want to minimize the difference of this from an established solution, if that makes sense. I want to minimize.
So exactly. So you don't.
So you're giving it. It's like the oil in the motor. It gives it a little bit more. It kind of makes it so that these things don't grade against each other.
Exactly. Yeah. So because one thing, I guess, is that, I mean, you won't say with 5 parameters you can fit an elephant, right? But that's not for these mechanistic models, right? That's
right. It's actually not true at all, right? Like I sometimes show this. It's a famous story, and it's a great story, and physicists especially eat it up, right? They love it. So I can talk about that for a second. I one time I knew I was going to get those comments, so I showed, somebody actually. There's this famous story where a student or somebody comes into, I think, Johnny von Neumann's, John von Neumann's lab, and says he's fit something, and he says, how many parameters did you use? And he says, well, 4. And he says, well, 4 parameters, I could fit an elephant. And with 5, I could make him wiggle his trunk, right? And sure enough, somebody out there has actually used 4 parameters to make kind of a little curve that looks like an elephant, and they can make the trunk move with the 5th, which is fun and exciting. But I think what the physicists are missing here, I show all of that in this talk that I gave, and then I just showed a picture of an actual elephant. Right. Just to be really clear, and if you said this in any other field, like imagine if you were trying to design a microprocessor, okay? And I said to you, well, if you use more than 4 parameters, it's BS, right? Like, they would just look at you like you're crazy. So, it's just kind of a little bit of a misconception. I'm not against small models at all, right? They're often the best way to look at a problem. I think of modeling as just thinking, right? Like, it's a way to think in a straightforward way. I'm totally in favor, just to be clear, right? I love the classic stuff. But this is something special that's going after something special. It would be overkill for certain problems, but it's essential for some problems, and those problems usually involve emergence and interactions between many of these different systems, which people typically study in isolation. So another way of saying this, one time a physicist, another theoretical physicist asked me, well, aren't you making a lot of assumptions to make this model? And I said, well, I'm making the same number of assumptions that you are, right? You're assuming almost nothing matters, and I'm assuming almost everything matters. But it's the same number of assumptions, right? We're both deciding whether or not to include these things. And for some cases, like your assumption will be right that you don't need to know all of this. But I'm looking specifically at the cases where it does matter, and so that's why I'm making a more comprehensive system.
I mean, obviously, we have the same kind of this plethora of models at different levels of complexity or simplicity also in neuroscience, which they play different roles. And well,
right.
I think you need all of them.
Yeah, that's exactly right. Like in fact, I mean, if we get a chance, I would love to talk about some of Timothy O'Leary's work at Cambridge and basically how I would think about modeling a neuron. It would be very, very interesting. I think it's an effort that's possible because neuroscience, I feel like biology is kind of the major people take if they don't want to learn math, but neuroscience is a major exception to that rule. They've really embraced to the point of AI, right? Like they've embraced it top to bottom. And so I think it's a really great place for theorists to start thinking about what could be included. When we think about spike trains at these very short time scales, what would happen if we started thinking about the regulatory changes that happen at slightly higher time scales? And a lot of methods exist to be able to represent those things. And anyway, we can talk about it later, but I'm excited about stuff like that.
Okay, so now we talked about the different functions of the cell, and now also how you sort of design sort of the overall structure of the model. And of course, now we're coming to experimental data. So I guess there's like a twofold use. First is sort of you need them to, well, determine like model parameters and also to test model predictions. So what kind of data did you use and can be used?
Yeah, that's really interesting. So we definitely used whatever omics we could find. In the mycoplasma model, one weakness of the model is that not that much has been measured, right? So we used a lot of other bacteria and mostly E. coli parameters, right? But we used single cell biophysics, like how molecules collide on nucleic acid. We used any kinetic parameters that represented how enzymes work. We used, expression data to figure out how much every gene was being expressed into mRNA, or protein. So we used half-lives and decay rates for the major molecules in the cell. So we pulled all that kind of stuff together, and that's how we did it. It is interesting, like right after we published this, we did get called by certain groups, like kind of demanding to know why we didn't use that data set, And it is funny, is a lot of data out there which really doesn't help. Like you can't put it into a model very easily. It doesn't mean it's useless, but I don't know if you ever saw this paper called how would a neurobiologist fix a microprocessor?
No, we had Konrad König was on the podcast.
Oh, really? That's a brilliant paper, and I think worth reading for anybody, including people who are not theoreticians, just because it sets the problem so well. their main conclusion was not only that they did not figure out how the microprocessor works, with a lot of things that totally did not really, they sounded plausible, but had no basis in reality at all. And then also, though, that the methods they were using would never lead them to be able to find. So I do think one of the functions of models can be to identify new experimental techniques that need to be developed. And we've done that, right? So the experimental techniques that we've pioneered were inspired by whole cell modeling, even though modeling does not show up in those papers.
Because I saw that this was quite heroic. I think you started with this like 2008 or 2009, and then you and your poor graduate students had to come through about 1,000 scientific articles for.
Yeah.
That was quite a Herculean task. I'm quite impressed.
E. coli is much worse.
Yeah, really. But that's fantastic. I mean, you really have to be stubborn.
Yeah,
And,
E. coli was much worse. I'll tell you just a funny thing there. So in the summers, I really like to do work. Like I like to, whether it's work at the bench or work on theory. And so I usually ask my team, like, what's the most disagreeable task that you have this summer? Like, how can I help? And one year for E. coli, they wanted me, we had identified 10,000 papers that had kinetic information in them about the enzymes in the metabolic network. And there's a database that houses all of those, but a paper came out, and it's called the Brenda database, but a paper came out saying that there were a number of errors, and so we had to double check. And so I vetted the 10,000, and then I read 1,200 papers that summer to get the metabolic parameters out.
Wow, that's a very impressive advice supervisor, I think.
But imagine, so the change, the error that happened, guess what error happened most often? The most common error translating those parameters to the database was mistaking a millif for a micro and vice versa. So it's not just like a random typo. It's like 1,000-fold error. So that's when we realized we had to do it all by hand, and it was painful. But yeah, now we have it.
You seem to be like a general that led from the front.
Yeah, right. You could see the wounds. You could see the battle wounds.
Exactly. You're sitting there with a... So then they sort of looked for, we're going through all this paper, looking for upper and lower ranges, I guess, or some like estimates.
Yeah.
Some to use in your... And then you also say, I think you said somewhere that, I mean, of course we have to say that all models are wrong, but some are useful. But you also say that all data are wrong, but some are useful.
Yeah.
I like that.
Do I dare say this in public? Yes, it's true. That's the funny thing. No experimentalist wants to admit this. When I love this in talks, because I'll say, all models are wrong, some are useful, and you can see all of the experimentalists nodding their heads somberly. Yes, he's right. This guy is full of it, right? And then I'll say, one thing people don't want to admit is that all data is also wrong, right? Like there's so what do you do in that case, right? You do cross-evaluation.
What do you mean that they're wrong because it's like experimental errors, or is some kind of...
Oh yeah, I could come up with a million reasons. In vitro versus in vivo, right? There often people have shown that the in vitro measurements can be off by many orders of magnitude from an in vivo representation, just because you have this crowded, at these extremely small concentrations, inside of a single cell, it can just be totally different situation, right? And then it could just be that the methodology is not quite worked out yet to test it. Whenever we're testing anything, or you added a fluorescent protein to another protein and now it can't move around as easily. All of those things are really, really important. And we're always making those approximations, right? We freeze the cells, we electroporate the cells, we use viruses to infect the cells so we can change the DNA. Whatever it is we do has an impact. And so, it's a little bit like, it's a little, kind of the more that you go into the system, the more you've probably changed or modified it by observing it. And so that's the key. That's why you need to have theory together with great experimental. So it can be compelling to say, oh, it's because we're training these students and they didn't know what they're doing. But I would say fundamentally it's wrong.
One thing, I mean, if you sort of do like, for example, model with these neuron models, biophysical detailed neuron models, you have some parameters that, for example, the amount of channel conductances that vary with time. And so you actually have, which are parameters in our models, not in your models, but and then so you actually have parameters that are not even constant in the over the time of interest. But I mean, your time of interest is about 10 hours or something like that. Do you also have problems that some parameters actually change with time during?
Yeah, right. Well, this is another way that everything's wrong, right? Constants are not constant.
Well, some are, I guess.
Yeah. Well, are they? Like, there's so much noise at the single cell level. There's like we use those constants, but they're abstractions that we're using to help us describe the world as human beings. So I do this exercise in class with my students where I show them raw data from gold standard labs, like truly outstanding raw data. And then we see, okay, you now fit this to a line, but does it really fit a line, right? Like we're assuming a line to help us, right? Even if we fit a curve, like these data are all over the place, right? And so then we say, okay, great, but we're going to assume that this is probably good. But then is it really good? Like, what's the range in which that's good enough? Or, getting a healthy sense of what goes into every number that you put in is really important, right? And to your point, I love what you were saying about conductances, because that goes right back to what I was talking about before. Like neurons, those conductances are achieved by the expression of channels, and the expression of channels happens on a time scale of minutes to hours, right? And it dramatically changed. But, wouldn't it be amazing to have a neuronal model? Maybe it's not a whole cell model, but it includes the expression of the channels,
I guess,
as a way of incorporating those.
That's what, like, essentially, I think, as you mentioned, Tim O'Leary has done some really cool work.
Yeah, that's what I mean. That's but
no,
that's what I see. If I were going to do a whole cell model of neurons, I would start by comprehensive gene expression and metabolism, which is not whole cell per se, but it would add a lot of really interesting details. So Tim and I have talked about this, and I'm a big fan of his.
I'm actually I have to invite him on the podcast, because I really, that was a paper I think he wrote together with Ian Marder's group.
Yeah. It's brilliant.
That's really, I remember that.
Yeah. We wanted to bring him to Stanford.
Okay.
But we were unsuccessful. He wanted to go back to England, which I think was a great choice for him.
It's especially now, maybe. Well, we won't talk about that.
Oh, that's a low blow.
Actually, sorry about that.
Here I am as your guest.
No, we all root for American science.
Yeah, perfect. And in the meantime, I'm currently waiting. I'm expecting to receive a German passport in the next month or two. So everybody's right. You're all rooting for American science. We're all wondering about European science.
Exactly. So you have some kind of German connections and German.
I grew up speaking German. My mom's German.
Okay. Well, you're most welcome back to Europe.
Yeah, thank you. Yeah.
So anyway, now we have this data. So now, okay, of course, you have some data that you get parameters that you have extracted from experimental papers, and now you have to start, some sense, fit the model, right? Because you have to sort of fit the parameters, at least the final tuning, at least some of them, right?
Yeah.
So and of course that's a whole new ball game, right? How to do this, and one sort of like the standard way, I guess, is sort of to run the models with certain parameter sets, and then look whether you get closer or not to your target, and then you jump around in parameter space and different kinds of methods, right?
Yeah.
So, but then that requires typically many runs of the model, right? So it typically become computationally expensive.
Yeah. Right. And to give you a sense of that, like the first whole cell model, it took a supercomputer basically to run one cell dividing one time, 10 hours.
Okay.
And so that took forever. And journalists at the time loved that because they're always like, oh, this guy's running simulations, but it takes longer to run a simulation than it does to grow this thing, right?
Yeah.
So that was really challenging. And I would say for this first one, not the E. coli one, but for the first one, that was a big limitation. And we were constantly arguing and trying to figure out how to set the parameters, because what we saw when you're trying to put the final thing together for the first time, the worst is that you change a parameter in one module and all of a sudden none of the other modules work, right? So you could get this individual to start working, but then it's not playing well. So that was very difficult, and we brought on a top flight software engineer to help us with that, actually. So this is where we're lucky to live in Silicon Valley, because I have access to some really great engineers who will come and do work here for a few months at a time.
So in order to speed up your simulations, essentially, or?
So we sped up. So the first thing we did was we had a test-driven programming scheme that they implemented, which I had never heard of before. But basically, he started us out writing tests of every single process, but then these inter-process tests. So they got more and more sophisticated, and you could see if it was passing any of them, and that would catch a lot of the problems earlier. So that was really important. And then for E. coli, we were really worried about this, but we had so that software engineer was one of the people who made Gmail profitable, right? So that's a pretty big achievement. Then he came work for us for a little bit. And then there's a guy, Jerry Morrison, who got us from, super long runs in E. coli to now you can run it on a laptop by your, in about 10 minutes.
Wow.
And he was part of the team who invented cut and paste, like Control C, Control V.
Oh, really?
And he put maps on the first iPhone. So he's just a superstar software engineer and helped us a lot. I think a lot of times people neglect this part of modeling, especially with these big models, that these days it's three legs of a stool, right? It's the biology and experiments, and then it's the modeling, but then it's also the implementation, which you have to think really carefully about how you implement stuff, or else it's just not feasible to do.
I guess it's also kind of like a skill set that is not always found in academic labs, right?
That's right. So I would say definitely in biology, and possibly even more broadly than that, like my team has probably received more training on this campus in like a big software design project than almost anybody, because they have these like mentors who've done such big projects, and they get a lot of code review and stuff like that, which is really it's a pretty invaluable training for them.
And then you also, I guess, you also arranged this sort of, or are involved in this dialogue for reverse engineering. And it's actually like this, some kind of competition that was set up so that people could, because I guess so you could try out different methods for optimization, because I sometimes say that in war and love and optimization, everything is allowed, sort of whatever you can.
That's right.
You can do whatever works.
That's right.
So it's very, of course, then you have like this wisdom of crowds. So that was sort of a quite nice move, I think.
Yeah, honestly, I wish we were doing that more. We only did that competition one time, and it was extraordinary, but there were definitely a few things we could have done better. Right now, I think we could do much better, because the E. coli model, I would say it passes my version of Turing's test, which is to say, if I generated, let's say that some experimentalist generated a set of experimental data of many different kinds, and then I ran a simulation to produce the same conditions of a data set, I don't think that there's an E. coli biologist who could tell them apart.
Oh,
Right?
that's interesting.
So it is interesting, because then you get into this phase where you could think about the organism as the simulated organism as real, right? And to this point about the neuroscientist and the microprocessor, if someone's trying to design new bioinformatics or processing methods, they should do it on my system, right? Not on real E. coli, because if they can't solve mine where the causation is known,
that's true.
then how can they solve it anywhere else? And the feedback loop is so fast. So I am a big believer that we should reconstitute some of those challenges. I think any method that you could invent right now to better understand my model would probably be a better method to use to understand a real biological system.
No, I think that's we are sort of using the same sort of to in neuroscience, like developing these data analysis methods, and we also to test them, to make sort of like this benchmarking data. But because the point is, I mean, if it should work, if it's a general method, it should also work on fairly reasonable sort of like benchmarking data made by the analogy of your wholesale model, for example.
Yeah.
So I think that's certainly something something that we should, as a field, pursue. Both there, both in your field and in like systems. Well, when I think of systems neuroscience, I often think about going from neurons to networks. There's like a lot of activity.
Yeah.
A lot of activity there. But also, so you said that when you started running your model, it was sort of crashed, and it's often, I think, when people start working with building network models, the network activity either dies or goes epileptic. That's sort of it takes a long time to get anything but that. So I think it's sort of analogous.
No, it got really tense toward the end of that first one, just because we knew we were so close, and yet running everything was taking forever. And actually, this is kind of fun. Most of the time, I don't have like an interview directly in here. Hang on one sec. Let me see if I can. Oh no, here it is.
Sure. Okay.
This is from the historical archive. No, this isn't it. Hang on one second.
Okay.
Okay.
Oh.
So we were trying to get this thing to run, and we knew we were so close. And then my student Jonathan, I had to go to a conference, and I just told him, if you find anything, just send me everything you've got, right? And so I get this FedEx at the conference. He finally got it to work, and it was this. He sent me this binder. He sent me this binder, and I had told him before, print out every single thing you can think of. So it's just a binder filled with plots from this simulation, because every simulation generates like a gig of, like, one cell divided one time generates a gig of data. And so I sat there trying to pay attention during this conference, but just like practically with my hands shaking, like, here I've got this thing filled with insight, I just don't know what the insight is. So very, very exciting moment in my career.
Yeah. Must have been a pretty good feeling when it finally worked out.
Yeah.
So, but because one important thing here also that I think you mentioned it is that the behavior of individual bacteria is stochastic, right? So, and the models are stochastic. So this thing of fitting to, so dealing with stochasticity. So you have to run some sense to run the model many times in order to get an overview of the predictor, sort of like population activity in some sense, right?
Yeah, that's right.
Okay, so now you have this really cool model, or it was sort of like, I guess it was published 2012, so maybe it was done in 2011. And of course, one thing you would like to do when you have this sort of this model that has passed all these tests is that you would like to look for new predictions. So if you sort of start with the predictions from the Mycoplasma model, so there were some new things that you could. I mean, you could sort of. I mean, obviously there were some new predictions that you made, right?
Yeah. So there were a few things that I think were particularly interesting in that model. One was the one that I just told you, right? That you have tens of thousands of collisions on the chromosome, right? People were not expecting that. And in fact, if all of those collisions lead to a displacement, the cell can't grow. It's too much. It like really, really hinders the cell.
So collisions, so that's like proteins
So like,
bumping
let's say
into.
let's say that the DNA replication protein is running, something else is bound here to try to get some gene to turn on, and this thing just runs through and bumps it up, right?
Oh,
Just
really?
like a freight train, yeah.
Oh.
But the problem is, again, this goes back to the data. What are the lies we tell ourselves when we make a model or when we look at data, right? So I would say in this case, what's interesting is when the collision experiments are done, single molecule biophysics or whatever, they're not representing it with a crowded environment. So they don't really know what's going to happen when a protein pops off. They just know that could make it come off. So, and now we have it's too bad. Like I remember it almost like yesterday because it was on this whiteboard when we were trying to figure it out. So, actually, some of Zan Schulten's data really helped me understand what we now call the crowded bar theory of how proteins can be removed from the DNA, but still it has no impact. So imagine that you and I are in a crowded bar, okay, and we go up to the bar, but there's tons of people there, and we order a drink. And so here I've got my drink in front of me, and I don't have an affinity to anyone else in the bar, right? Just my drink, right? But then someone has to use the restroom, so they come by and they push me away from my drink, right?
Okay.
So yes, that bond is broken.
Okay.
But there's a lot of pressure from everybody else because it's so full. I'm not going anywhere, and I only have affinity to my drink. So of course I'm going to end up right back at that drink momentarily, right? And so if you think about E. coli transcription factors or other proteins that bind DNA as like in the crowded bar with one affinity and not much else, and like a lot of pressure, then that can kind of help you understand how this could be the case, that you could have all these collisions and yet the proteins are able to stay where they are and complete their function. I know there's no proof really for the crowded bar theory. We have lots of dumb name theories like that, which I'm happy to share at any time. Maybe in a crowded bar someday we can cover all of these, right?
Exactly. Yeah. So and one thing I think you there was a paper in 2013 where you sort of showed this like mutant strains, and you could sort of see how you could sort of predict sort of how the different mutants, like how they grew or the effects on like the growth rate, and that seemed to work okay for all these different strains.
Yeah. Thanks for reading that one. I feel like that's the most unappreciated paper that we've ever written, but it's super, super important. Like that goes together with the original paper because, and I'll take you through why. So Craig Venter, like at the Venter Institute, John Glass was generous enough to let me come to the institute and learn how to grow these mycoplasmas, right? And so I learned that, and I came back with a knockout library, well, like a transposon library where these genes had been disrupted, and so I could actually test firsthand the essential and non-essential genes, and which was amazing. And I was so excited. We found that we were in most cases able to predict not only whether or not a deletion was essential, but also predict quantitatively what would happen if that gene was removed, what would happen to the growth rate. That sounds amazing, but in reality, it was extremely disappointing because this cell is so fragile that it basically is binary. It either lives or dies. There's only a couple genes at all where, and literally a couple, maybe two or maybe three, where it has a real impact on the growth rate but still grows. And so, but what became more interesting was that in some of those cases, we could actually start to predict more deeply about the enzyme, like the chemical or the kinetic parameters of the enzymes that created those growth behaviors. That's the real amazing thing here. And the way that I look at it is, if you think about like Lawrence and chaos and this idea that a butterfly flaps its wings, in Oslo, and we have a hurricane over here in San Francisco. In some ways, what you could argue that we're trying to do is take all the butterflies and everything else that's in, creating the behavior of a cell and include it and see if we can predict the weather patterns, right? But what Jayadita decided to do, she was the lead author on this. She thought, well, if we know the weather patterns, can I go back and predict how fast a butterfly is flapping? So she saw these growth rates and she said, oh, maybe this can give me insight into some of the kinetic parameters of enzymes that haven't been adequately measured. And so the highlight of that paper, I mean, really, it's I would say the last figure of that paper, Jayadita went in and for one of those enzymes, and I was like, I don't think this is going to work, right? I don't think we'll be able to do this. But Jayadita was able to show that that she measured a kinetic parameter that the model predicted correctly, and that was orders of magnitude different from what had been thought to be the value, right? And I told her, even at that moment, I said, go get your postdoc right now, because that'll never work again. I was just like, you did it, just go get your job. And then, but she did the replicates, and then she did it for 2 more proteins. So we have 3 cases in there. And that was the first time I realized that, wow, maybe this is more than just like an achievement of technology. Maybe this is really a biological engine that can drive discovery. And that's turned out to be... Go ahead.
No, I mean, it really sounds like a non-trivial prediction that is sort of validated or tested. And and then, so this model is much more than just something that fits the training data.
Yeah, that's right.
It captured much more.
Yeah. Jayadita really had the courage to do that for the first time, right? Like to really go out on a limb, like, let's push this as hard as we can. And of course, it doesn't always work, right? But when we find something surprising, my first assumption is still that it's a bug, right? But I will say that those predictions are highly enriched for new biology. And especially in E. coli now, I've become more and more confident and trusting that it's something real. And also I'd say she set an example for just trying for a bigger, trying for something bigger, trying for something more ambitious in the predictions and how we test them.
Because I guess when you make this kind of wholesale model, there's sort of like one thing, if it's it can be successful in predicting sort of the behavior of the whole model and the wholesale model and the new circumstances. But it can also be successful in actually predicting the underlying parameters, meaning that the physical process of the parameters, and that's 2 different things, right?
Yeah, that's right.
Yeah, and
yeah, I agree. We talk about this a lot. Like I also think one thing that Jayadita did is kind of refocus us, because a lot of people say, we want you to predict the growth rate of something, and that can be really important if you make perturbations, et cetera. But usually I tell them, the growth rate is almost the easiest thing to measure. So why not predict something harder, use the growth rate as a parameter, and predict something more hidden from us, like, right, more obscured from our view. And that is also powerful, right? The fact of just like, let's use the easiest measurements that we can make. Let's use them. So yeah, that's use them to springboard something harder.
And sort of is, I mean, if you sort of look at maybe the coolest model in cosmology, right, with this dark matter, dark energy, it's all based on sort of going from these cosmological observations and then inferring back to these parameters. So in some sense, this is when it works, it's really amazing.
Yeah.
So, okay, now to E. coli, right? And then, so this was something that I guess you started on, well, I don't know, after your mycoplasma. And then you had the paper in 2020, I think.
Yeah.
And why E. coli?
Yeah. Okay, so the mycoplasma model, this paper with Jayodita, it was amazing because of what she did, but it was very disappointing otherwise because it just can only grow in one condition. It's not very complicated. So I knew I wanted to do something bigger, and E. coli made the most sense because
Okay.
it's vastly more complicated. It's got all this regulation. It can grow on almost anything. It's valuable in its own right. So, a place like Genentech, for example, they have 3,000 different production strains that they use to, to make insulin or whatever else they're making. And they, it's always a challenge for them to know which one should we pick, like how should we optimize this. So it has industrial value, it has basic science value. CRISPR was discovered in E. coli, right? So I keep telling the lab there are at least 10 more Nobel Prizes hiding in E. coli, right? We should be looking for very, very, the reason why people haven't found them is they're more complicated, they're more emergent, right? So it's not about chasing a prize, but it's a way to set your sights higher, right? We don't want to predict a number anymore. We want to predict a phenomenon. And so we've really pushed hard in that space, and it's been incredible. Like, this coming year is going to be a golden year for whole cell modeling because the E. coli model has led us to several super exciting new observations and discoveries that we're proving experimentally and that have just been completely a joy to look at and work on. So
excellent. Because you said that, I mean, it's, of course, it's because it's easier to study experimentally, and it's so much experiments already done. So
yeah,
I guess it was in principle easier, maybe more information to constrain model parameters, even though it was like 20,000 of them.
yeah.
Yeah.
No,
So,
it's harder. It's easier in ways that I like, and it's harder in ways that would be necessary anyway. So it's easier in the sense that that, like you said, we have access to parameters. All of our parameters that we use come from E. coli, unlike our previous model, right? If we have questions, we more often than not can actually go in and answer them. It's harder in the sense that it can grow in almost anything. It's harder in the sense that these cells are so heterogeneous that you have to think about the colony level, right? Like, you have to think. But, when I think about, my dream would be to simulate a tumor someday, right? And same thing, you need heterogeneous cells, you need different environments, you need that complexity. And all of those things, even though we're not working in a mammalian cell, we're developing methodology that would be more or less directly transferable.
But this, I think I think I read in your paper is like 10,000 equations or something for E. coli cell. So how comfortable can you run this on, is like normal computers or
like I said, our software engineering team has gotten it down to about 10 minutes on a laptop for one cell.
That sounds like magic.
Yeah, it does. I agree, but it's very complicated. I would say the bottleneck right now, interestingly, and this is true in a lot of computational things, right? One of the bottlenecks is just accessing all the data we generate, right? So that's like a problem of computer sophistication and just being able to access data. And then another limitation is, honestly, the model has become so complicated that figuring out what it's doing is hard, right? So we'll run a bunch of sims and then we'll sit around for weeks and months trying to think, what in the world does this mean, right?
But it's sort of,
So it's still faster than the biologist, but I would really love to investigate better ways. Maybe AI, everybody says AI is the answer to all their problems, but maybe AI, maybe other more sophisticated statistics to try to draw out what's going on in these. Like, how do you analyze a gig's worth of data, when it's also heterogeneous? And
but it's also, I think some people call it the white box, because in principle you could look into it, right? and know all the parameters. So
totally.
it's just that
But it comes together like very often at this point. We see phenomena that we did not include and that do not look like. It's just a very nonlinear. It's so nonlinear at this point that you can look at it and just think, where in the world did that come from? That's what will make this next year so special, is that you see these things that you just wouldn't expect, and then they lead you to things that you would not.
Okay, and there's also these unexpected things are seen in experiments afterwards, or?
Yes. But then we finally have we make the explanation in the model, and then we try kind of rabbit out of a hat type of experiment that no one would ever do, because they would think, why would you do something so stupid? And then it works. So I can give you one example. Like, we've been studying metals a lot lately. Metals, because metals are important parts of enzymes, right? Cofactors of enzymes that enable you to catalyze reactions.
Okay, yeah.
And they're kind of understudied. Like, usually when you even put metals, you don't usually, when you do a test of E. coli, you don't even usually put metals in the media. You just use the trace metals that are in the glassware, right?
Okay.
And but too much is toxic. Too little is not enough to support all the enzyme content. And it's an important part, as you know, like zinc supplements or whatever. It's an important part of the immune system. Like, our bacteria in our bodies are constantly fighting with us for iron, for example. And it turns out there are massive trade-offs of the different metals that are used to support growth, to support. So I'll tell you a couple things. I mean, so the biggest thing I can tell you is that we've found out that a lot of trade-offs happen. We've been able to simulate the metalloproteome of E. coli under many different conditions, and it turns out sometimes there are enzymes competing for these metals that both are required for growth to happen optimally. And so if you do targeted supplements, you can make them grow faster. And so for one, I think we have I'm not sure. So if anyone listening thinks they have a faster, we have an E. coli right now that with the right supplement of metals can grow almost as fast without oxygen as with oxygen.
Wow.
And it's just by metal addition. And so I think it's a Guinness Book. I was looking through the Guinness Book of World Records. It turns out nobody cares how fast bacteria grow, so we're not going to apply for a world record. But I think this is something. Just imagine fermentative growth. If you can make it grow so much faster, that's worth money for sure. So that's the kind of stuff that you would never expect a model to tell you. Because we didn't, so it's an emergent phenomenon that arises once
it's
you've
sort of
covered
what you
all that
hope
stuff.
for at some point that the models will do, right? Like the Maxwell equations that predicted light and how to send radio waves, right?
Yeah,
that was unexpected, and that worked. So it just tells something about the quality of the model, I guess, and how well it is. So that should be.
And it will never be perfect. It will keep becoming more and more complete. But my hope is that it will occasionally give us these gems of insight that are, really worth something, to the world. And I feel like that's kind of more or less where things are going. So that's been really exciting.
One thing you also found is that most sort of all this E. coli data were consistent with each other, which sort of was a good sign for the field, like a relief. And you also introduced this term deep curation.
Yeah.
Well, what do you mean by that?
Yeah, deep curation is really interesting. Like, if okay, so the general idea of deep curation is this, right? So if you think about deep learning, the idea is that you're looking at interactions between things, but you're also looking at the interactions between the interactions, right? That's deep learning in a machine learning type context. And so we also think about layers and interactions between interactions. So if you think about data layers, So maybe I'll tell you a story first, because this is where the idea came from. So when someone was reviewing the original mycoplasma model, we said we had validated it using a couple of data sets, right? And the reviewer had this, they must have been some kind of a philosophical theorist. They
Okay.
said actually they haven't validated this model because it's impossible to validate the model, because every piece of data is actually linked to everything else. So there's no such thing as an independent data set. As a result, it's impossible to independently validate it, which means it can't be validated. And
Wow.
so they didn't validate it. So
No.
so that's fascinating, right? I could disagree with that in some ways, but that's kind of an engineer talking to a pure theorist. But it made me think a lot. Really, every piece of data is linked to every other piece, but it's linked deeply. That's the key I want you to see. So let's say that we have this data layer where we just put all the data that anyone's ever generated in E. coli, and it goes from like, an RNA-seq data set that counts all the RNA in the cell, that was generated last week, all the way to 50 years ago when someone spent their entire PhD trying to carefully measure one biochemical parameter for their thesis, right?
Yeah.
And so those things can't be linked. Right now, you can't really evaluate them against each other, right? They're totally separated, and they might be in totally different parts of even the model. But from the raw data, people have made a parameter layer, right? Maybe they fit all of their measurements to a line, and they got a kinetic parameter out of it, right? Michaelis-Menten parameters or whatever. So then you have this parameter layer, which takes a bunch of the raw data, kind of encapsulates it, and represents it as a parameter. And then you have further an equation layer that you have to curate. So we typically curate the raw data and the parameters by storing them, right? And then you have an equation parameter that's interesting because that represents mechanistic knowledge. That's what typically AI does not have, right? They don't have an equation layer that's really holding together like the mechanism of how we think things work, like Hodgkin-Huxley or Michaelis-Menten, right? You're assuming a mechanism there. But the equation layer takes all the parameters, right? And then we put those into modules, and then we put those modules together in the whole cell model. And now all of a sudden, everything is linked, but deeply. So if you take that RNA set, you now can actually directly compare it to that measurement that somebody made 50 years ago. You just have to go down the layers into the model and back up the layers, and you can see how consistent they are with each other in the sim. That's deep curation, and it's kind of a crazy, I'm telling you, even when I'm telling you, I think it's a crazy concept, but that's how we do it, right? We add in new structures, and then we do this deep curation, trying to see where the data fit against each other and where they don't. And the reason why we're looking for where they don't is that's exactly what the guy did who discovered Neptune, right? The orbits don't fit, so we're looking for the point of friction. And that is probably where new biology is hiding.
Yeah. So, no, this is, I think this, of course, me as trained as a physicist, I'm still doing physics, I would say, even though I work on the neuroscience problems. This is sort of the kind of, this is my ilk, this is kind of the culture I come from. So, but because one thing that I think this computational biology, the culture of it, in biology, I think you mentioned it in, maybe it's you wrote a very nice book in 2015, which I recommend. So I have it here. And you mentioned something about, like in the sidebar, that you need to do some experiments to get some credibility in the biology community. So what's your sort of cultural experience of doing this modeling, which is sort of like a quite sort of like new way of doing science in this branch of science, in your field, right?
It is tough, right? It's because we are kind of crossing this bridge, excuse me. It's a little tough to, the good news is that the younger generation gets this totally, right? Like they understand that this is needed. And I would say, well, Here, hang on one sec. I would say that I can best express it with a joke that's actually in the book that I first heard, I think, from Lee Hood, who was one of the big sequence pioneers, you know. So there's this computational biologist, excuse me, computational biologist who's driving through the countryside and sees a flock of sheep and thinks, wow, I'd love to have one of those. So they jump out and they say to the shepherd, hey, if I could tell you how many sheep you have there, can I have one of them? And the shepherd says, oh, yeah, that'd be amazing. And they say, yeah, well, you have 964 sheep. And they say, wow, that's amazing. Yeah, go ahead and pick one out. So they grab one and start bringing it back to their car. And the shepherd says, wait, if I can tell you what you do for a living, can I have it back? And he says, yeah, sure. So, well, you must be a computational biologist. They say, wow, yeah, but how did you know? And they said, well, that's actually my dog you're holding. So we have to be really careful, right? Like we have to be careful that we are not holding the dog. Because so often
it's embarrassing.
Yeah, it's embarrassing for everybody, right? It's embarrassing for the shepherd and it's embarrassing for the computational. So I think in some ways, by focusing on the wrong things, we have lost some credibility with biologists. Or, you know, the younger generation, I still think they get it. They want more of it. They understand that this is valuable. But we have to make sure that we're not, you know, in Silicon Valley right now, there's almost like this feeling like, you know, I'm ignorant about this field and that's my superpower. You know, I can disrupt this thing, right? I can go in and disrupt everything, and and that'll be the way we figure it out. No, expertise still matters. Knowledge still matters a lot, right? And even for these AI models, right, they're based on data, and people always say data like it just magically appears, but that data is like the life's work of thousands and thousands of people who have lived and died to bring that to the world, right? And that's what AI is based on, not just on a method of, you know, a bilinear correlations. So I think that it's really important that we stay rooted in just this deep knowledge of what we're studying, and I don't think there's a substitute for that, you know, right now at least.
Absolutely. I actually read this, you know, John Hopfield obviously got the Nobel Prize last year.
Sure.
And he wrote this article about, because before going into neuroscience, he was working on this biochemistry problems. And he wrote this actually it's quite fun. I'll make some links to your papers. This paper on the
I'd love to see it.
Yeah. But anyway, he said that he was at this conference in Italy somewhere, and he said that, I just read this, one of the senior biochemists took me aside to explain to me why I had no place in biology. As he said, gentlemen did not interpret all the gentlemen's data, and preferably worked on different organisms. If you wish to interpret data, you must get your own. And also went on to say biology is very complicated, and any theory of mathematics is such an oversimplification that it's essentially wrong and thus useless. And then, but then the advice from Hopfield is, well, you should stick to the physics paradigm if you're already in it, and have a thick skin. So, have you needed a thick skin?
I honestly feel like, so definitely plenty of people have skepticism, and I encourage it, right? Of course, we're scientists, right? If I can't be a big kid about, you know, people being skeptical. But one thing that I do take pride in is that the more people know about it, the more they recognize how powerful it is. So it's easy to be, you know, a 4-parameter elephant kind of person. But I mean, I think you could kind of, almost everything Hopfield has said relates in that story. I mean, these days we believe in big teams. Like, we know the value of big multidisciplinary teams, right? We know that there are so many things going on. We have the computing power to be able to represent things that prior weren't I'm sure it was almost like a dirty word or an indecency to use a computer to do some of this stuff. But of course, I'm sure even the great polymaths, they would not begrudge us the opportunity to use these incredible tools. Maybe the most exciting time I've had pushback ever, before we published the mycoplasma model, I really wanted to make sure that it was, because if you publish a high profile paper like that, and it's wrong, it's almost like an advertisement to the world saying, hey, I'm clueless, right? Like, you don't want to do that. And so I strategically chose two places where I talked about it before we submitted it for publication. And both of them were career highlights. First, it was a Harvard theory lunch. Those are amazing. I can do a plug for them. People get invited to do a theory lunch at the Harvard Systems Biology Department, and it's all whiteboard. So you're not allowed to bring slides. So I went with whiteboard only to talk about a whole cell model no one had ever heard of. And maybe I'll show you one more fun artifact. Hang on.
Sure.
I did want to show them a simulation. So I had these flip books made. So instead of showing it on a screen, you can actually see you can actually see a simulation in this flip book. So I did this theory lunch, and I really wanted to see what people thought there, and I got great feedback there from some amazing heroes of mine. So that was great. But then the second one was, because I knew about this Project K, I went to see Sydney Brenner, and Bernard Paulson had set up this meeting in La Jolla where he lived, where he came with fire. Like, he was ready to test me on everything about how the model worked. And for hours we talked, and I would say at the time, he probably knew better than almost anybody. There's some amazing people in Denmark who also are very, very good in E. coli. But in America, in the United States at that time, I would say there's probably nobody better I could have talked to about it. And afterwards, he kind of gave it the nod, like, you can go ahead with this. And so anyway, I love those kinds of discussions, because that's how we're going to learn. That's how we're going to get better. And I definitely do not mind at all. The beauty of being scientists, I feel like we're kind of the last great idealists. Here we are, you and I, two different countries, having a really engaging discussion about crossing our disciplines. And somehow this is like our job, right? Like, this is what we get to do for a living.
The privilege of being a scientist is
totally. And anytime you can have a positive argument, right? Like about stuff like that, not like the dismissive thing that Hopfield's describing, but like a positive debate, even if it's challenging. I mean, I say it's better.
Also this idea of, because I think when you come from physics, when you have you have shared Nobel Prizes between people doing the experiments and people developing and finding a theory to explain it, right? And in biology, and I know neuroscience, and sometimes this idea that I make all this data, which of course is a big effort and requires a lot of skills, and someone else finds something in the data which I didn't see, is it should be something to celebrate, right? Because at least that I only been doing sort of like modeling and physics and simulation, and it's not that easy. I sort of worked hard to learn these things. So so they should appreciate that too.
Right. No, it's great that you even say it's not that easy. No, it's not easy. In fact, it's not easy at all.
No.
Crick, of course, Francis Crick is the most famous of this from our field, right? He used to drive everybody crazy because he would understand their experiments better than they would, But that doesn't mean he was stupid, right? He was brilliant, right? And same thing. I would welcome the opportunity to work with somebody who could interpret what we were doing better than what we do.
It's partially, but I also think I've been in neuroscience now for like 20 years or something, 25 years, and it certainly is a change between the older generation and it's like so I think it's and also I think Brenner said that Francis Crick was the only one allowed to do modeling or like theorizing because he had sort of earned his this do.
Mm-hmm.
So that's also a thing. So I mean, this model and that I mean the E. coli model. How can the community use it in a simple way, or?
Yeah, it's all available on GitHub, and in fact, I consider us hyper transparent. So usually, in almost all cases, everything we did yesterday, you can get today. So we're not hiding anything. There will be we're going to for some jobs that we do lately, there might be a little bit more more of a delay than that, but not much more. We are always going to make everything available as early as we possibly can.
Excellent, yeah, and also so that people can. I mean, obviously, I mean, we talked about Allen Institute, and they are sort of also not only making things available, but they're also making it available in a form so that it's normal people are able to.
Yeah, it's Python. And I should also say about the book, the latest thing with the book is that we made a companion to the textbook that's essentially has everything the textbook has, but it's all Python notebooks.
Okay.
So
excellent.
You can actually change and it's free.
Yeah,
but you don't need to buy it. That's not a. I appreciate that, but the fun part that I should send you a link to is the fact that nowadays anybody can mess with the figures in Python and basically get all that knowledge for open and online. So I really believe in making things totally online for people. And hopefully, do that with the sim. And also, I will say for people who are serious, if you're out there and you're serious about it, we've had lots of guests who, they'll come and visit, and we'll help them get started so they can use it. Mm-hmm.
Yeah, at the Stanford campus.
Yeah, so that's right. At the beautiful sunshine. Here I'm sitting in the shade of ginkgo trees.
Yeah, no, I think it sounds very tempting.
Yeah.
So for most people, I think so. That shouldn't be. So I mean, so now the plan. I mean, you're still going to work on E. coli, and then maybe to some because that's still a bacteria, and then maybe some eukaryotic cell. You mentioned yeast, or you're not so sure, or?
Well, I really I've talked to Tim and a number of others, and I love the idea of doing a beta adrenergic neuron. I love it. I don't know anything about it.
What is a neuron? I know beta.
Oh, it's just a certain type of neuron that people usually study in culture.
Okay, so that's why it's
Mm-hmm. And so I think a lot of the parameters would be available, and you could kind of work with it. I would only do that if I had a greater consortium of people who really wanted to do it and thought it would be exciting. But I love the idea, and I think like I was just saying, thinking about expression is the first thing you could do, and then thinking about metabolism, even if you're not using a full-blown whole cell model, you could do some real damage. I think understanding some of those higher time phenomena. So that's my thinking. And I used to spend a lot of time at the Allen Institute because Paul Allen generously funded our lab for many years. So that's when I hung out with Christoph and all those guys. Anton,
absolutely. No, these
fantastic are scientists.
Absolutely. And so Christoph is quite often now over in Norway because his
is that right?
His daughter works there at the University of Oslo.
Oh, that's fun. I'm actually going to be nearby to you in a few weeks because there's a Nobel Symposium in Stockholm.
Okay. So you're most welcome to come to Oslo and hang out.
Yeah. Maybe I'd love coming to Europe when I get a chance. This one's pretty quick, but I'll let you know next time I'm in the neighborhood because I definitely want to go see some people up there.
Yeah. So now and then you talk about back to your plants, so tumors. I mean, a tumor, because that's essentially like similar cells but not identical.
Yeah,
because something like C. elegans, that's too much.
I mean, I think C. elegans would be amazing. I'm hoping what we do will kind of catalyze more of a field. I would love to see this become less of our lab and more of a field where people can do this, and anything I could do to share, what we've been able to do. But what is it 300 neurons in C. elegans, right? And how
I
many
think
cell
I think
types?
it's 900, but it's like 300 in his nervous system or something.
Yeah, right. So
Yeah,
it's not,
yeah.
and then a ton of the rest, I'm sure, are muscle.
I have no
So,
idea. Yeah,
so it can't be.
yeah.
Right.
I guess
I
also.
love that
Yeah,
it's finite, right? That's the
yeah,
incredible. I
yeah.
think people have talked about virtual worm before, but I think going in a multi-scale way where you're talking
Yeah.
about molecules to worm, that's pretty cool.
That would be.
Yeah.
Well, I mean, it's going to happen sometime.
Yeah,
I'm
that's
not quite
right.
sure. Yeah, that's for sure, right?
Yeah.
And industry is interested in this.
Yeah, I would say lots of industrial efforts are kind of in this
Yeah,
area, and but I would say for most people right now, it's premature, right? So if they just want to start using the whole cell model, but DARPA, we are currently negotiating with DARPA on a project that would force us to be very industry minded, which is to say, uh, they actually are even hiring a validation team where we would be expected to uh, make protein or molecules in E. coli that are greater than the state of the art for metabolic engineering
okay.
using model designs. So that's
So so then
very
using E.
interesting.
coli to produce particular molecules, is that sort of like
Yeah, at a
the
higher yield
yeah?
than had
I
been
see.
possible without the whole cell
Huh.
model. So that
Okay.
sounds very challenging to me, right? It's a
Mm,
grand challenge type project, but I think but if it works, I think that will open up the industrial possibilities by a lot.
mm. And finally, about sort of like the AI sort of. I mean, one thing is, I just sort of we had a podcast with Andreas Tolle, also just had this. Well, he's a colleague of you right
Mm-hmm.
now at Stanford. So he had this very nice foundation model for optical responses to in mouse visual cortex, right?
Mm-hmm.
So he essentially has trained this model to predict all kinds of responses
Yeah.
of these mouse neurons. And I guess you could also make a foundation model for your E. coli, right? So that you could sort of.
Yeah.
I'm not
So
quite sure if
people are
that's.
talking a lot about virtual cells over here, right? Virtual
Yeah,
cells, foundation models for
yeah,
virtual cells. Um, I think these are really, the jury is still out what's
yeah.
possible, right?
Mm,
And black box versus white box is kind of foremost in my mind.
mm,
Another thing, there are a few things that I think, I personally think that both things will be,
mm,
kind of required. I'm just looking at a note here. I wrote a little piece about what I thought were some of the advantages of this approach, right? I think the disadvantages are it's hard to make them. It's it's going to be much easier to make a foundation model of a mammalian cell, for example, just taking in everything. But the advantages that I think we have on our side, and in the end, I think we need hybrid models, right? Of course,
mm.
I was going to say that because my whole career is based on these hybrid models anyway, right?
Oh,
Why not use
yeah,
everything?
yeah.
But the advantages that I think a mechanistic model has that are really important, one is that causality is explicitly embedded, which
Mm-hmm,
means that the predictions are explainable, right?
mm-hmm,
It's also important to realize that mechanistic models learn in a different way, which really requires less data, probably. And then finally, the knowledge transfer learning is based on evolution as opposed to traditional transfer learning approaches. So there are certain interesting differences that I think are important to explore and probably in the future.
cool. I think this been almost a couple of hours, so I think
Yeah.
I used up my allocated 2 hours. So I really appreciate this. I learned a lot. So I don't know if you have any particular questions, something we didn't cover, something you would like to say in the end.
Well, I have lots of questions about how you do. I was thinking I should start a podcast just so I can hear what he's up
That's the
to.
best.
But
That's the best way to learn
yeah,
is to make a
I bet
podcast.
that's true. But
That's
no, I
good.
really enjoyed it, and to anybody who wants to talk more, you should just know I'm always happy to talk. I really enjoy engaging with the
Yeah,
community. Again, I feel
yeah,
like that's the luxury of our profession.
yeah. No, I think personally, I think sort of like your boldness is doing this. I mean, while people sort of discussing why this can't be done, it's too complicated, too many parameters, and you just try out. I really like that. That's sort of like the pioneering spirit. So
Thanks.
I think I'm very impressed by that. So,
I appreciate it.
Thanks a lot, Markus.
Thank you. It's a pleasure to be on here. I really appreciate the opportunity.